Okay, let's unpack this. If we think of our DNA as like the ultimate operating manual for our bodies, today we're diving deep into some of the classic errors you find in that manual, the predictable patterns of single gene disorders.
Right. We're talking about conditions caused by just one faulty gene, but one that has, a really big impact, not those super complex diseases with loads of genes involved.
Exactly. These follow rules that Gregor Mendel figured out, 150 years ago? And understanding these rules is just fundamental.
It really is.
And here's something wild right off the bat. We focus on these disorders, but actually, every single one of us listening, we're all carriers. Carriers for several potentially harmful genes. Mostly recessive, hidden away, which is why we're generally okay. But that potential is always there.
That's exactly it. And that's why this stuff matters so much. It's not just academic. This is the framework genetic counselors use every day. It helps predict risk, understand how severe something might be.
So what are we covering today? The big ones.
Yeah, the big three: autosomal dominant, autosomal recessive, and the X-linked patterns. Knowing these really unlocks a lot.
Okay, but first, quick clarification. People throw around hereditary and congenital like they're the same thing. They're not.
Definitely not.
Huh?
Hereditary means it's passed down, parent to child through the genes, inherited.
Got it.
Congenital just means you're born with it. Could be genetic, could be something that happened during pregnancy.
So a genetic disease might not show up at birth.
Exactly. Think about Huntington disease.
Huh.
It's absolutely hereditary, passed down through families. But the symptoms, they usually don't start until someone's in their 20s, 30s, sometimes even later. So hereditary, yes. Congenital, no.
That distinction matters. Okay, let's tackle the first pattern. Autosomal dominant AD. This one feels pretty direct.
It does. The basic rule is pretty simple. You only need one copy of the mutated gene to actually have the disorder. Being heterozygous is enough.
And autosomal means it's not on the X or Y chromosome.
Correct. So it affects males and females equally. They can both get it. They can both pass it on.
And the odds, if one parent has it...
If one parent is affected and the other isn't, then for every single pregnancy, there's a 50% chance the child will inherit that faulty gene and have the disorder. Like flipping a coin.
50%. Seems clear. But here's where it gets really interesting, right? Because it doesn't always look that clean in real life. What if someone has the gene but seems totally fine? Doesn't that break the dominant rule?
Yeah, it feels like it should. But that's where we get into some crucial nuances. The first big one is incomplete penetrance.
Penetrance. Okay.
I mean, someone inherits the gene, the mutation is definitely there, but they just don't show the trait. The phenotype, the physical signs aren't expressed.
So they have the gene but no symptoms.
Exactly. Maybe a disorder has, say, 50% penetrance. That means half the people carrying that specific mutation. They look completely normal. They might not even know they have it.
But they can still pass it on.
Oh, absolutely. That's the tricky part.
Okay, so that's incomplete penetrance. What else complicates it?
The other key idea is variable expressivity. Now here, everyone who has the gene does show the trait, but the severity is all over the place.
Ah, so it always shows up, but how bad it is varies?
Precisely. The classic example is neurofibromatosis type 1, NF1. You could have one person in a family with just a few kind of harmless brownish skin spots, those cafe au lait spots.
Right.
But another family member, same gene mutation, might have, hundreds of skin tumors, maybe learning issues, serious bone problems, huge difference in severity.
Wow, same mutation, such different outcomes. How do doctors even know it's the same thing?
Well, nowadays, genetic testing helps confirm it. But understanding why this happens goes back to the type of protein affected. And this is key for AD disorders. They usually mess with structural proteins or regulatory proteins, not typically enzymes.
Okay, why that difference? Structural versus enzyme.
Well, think about structural proteins. They're like the building materials of the cell, the girders, the cables, the membrane.
Right, the scaffolding.
Exactly. Now, if you only make 50% of the normal amount of a crucial building block, maybe for your red blood cell membrane, that 50% reduction can be enough to weaken the whole structure.
Makes sense, like building a bridge with half the steel beams.
Kind of like that, yeah. That's what happens in hereditary spherocytosis. A defect in a structural protein called spectrin makes red blood cells fragile. Just having one good copy isn't enough.
Okay, so a 50% reduction is sometimes enough to cause problems. What else?
Then there's something even more dramatic called the dominant negative effect. This is where the bad protein doesn't just fail to do its job, it actively messes up the good protein made by the normal gene copy.
So the faulty one interferes?
Yes. Think about collagen, the main protein in connective tissue.
Yeah.
It normally forms a triple helix, like 3 strands braided together.
Okay.
If one of your gene copies makes a faulty collagen strand, that single bad strand can prevent the whole 3-part rope from forming correctly. It basically poisons the assembly process.
So it's not just missing one good strand, the bad one ruins the whole thing.
Exactly. The result isn't just like 67% function. It might be close to 0 function. This happens in some severe forms of osteogenesis imperfecta, brittle bone disease. The defective protein is actually worse than having no protein from that allele.
That's a powerful effect.
Yeah.
Dominant, negative.
And there's one more main mechanism, though it's a bit less common, gain-of-function mutations.
Gain of function. So the mutation makes the protein do something new.
Yeah, something it's not supposed to do, and often something toxic. Huntington disease is the classic example here. The mutated Huntington protein doesn't just fail at its normal job. It gains a new toxic property that actually kills nerve cells.
Even when the other gene copy is making normal protein.
Even then, the toxic effect wins out.
It's also worth mentioning, isn't it, that a lot of the severe dominant disorders pop up as new mutations, especially linked to older fathers.
That's a really important point. If a dominant disorder severely affects someone's health and ability to have children, the gene might die out in that family line. So, for the disorder to persist in the population, it often needs to arise again through a new mutation, a spontaneous change in the sperm or egg. And there's evidence linking increased paternal age to a higher risk for these de novo mutations.
Okay, that covers dominant disorders really well. Let's switch gears to the biggest group, autosomal recessive, AR. If dominant was often about structure, you said recessive is more about chemistry.
That's a great way to put it. Yeah, AR disorders typically happen when both copies of a gene are mutated. The person is homozygous for the mutation.
Then the parents.
The parents are usually carriers, heterozygous. They have one bad copy and one good copy, so they typically don't show any symptoms.
So the risk for their child is different.
Right.
Yeah.
With each pregnancy, there's a 25% chance the child inherits both mutated copies, one from each parent, and has the disorder. There's a 50% chance the child is a carrier like the parents, and a 25% chance the child inherits 2 normal copies.
And this category, AR, includes almost all those inborn errors of metabolism, things like lysosomal storage diseases.
Exactly. Because AR disorders very often involve genes that code for enzymes. Enzymes are the catalysts, the workers that drive all the chemical reactions in our cells.
So why are carriers usually fine if they only have 50% of the normal enzyme amount?
It comes down to what's often called a margin of safety. For most enzymes, having just 50% of the normal activity is actually enough to keep the relevant metabolic pathway running okay.
Huh. So the body has a lot of backup capacity there.
It really does. You usually don't get a metabolic traffic jam or a buildup of toxic stuff unless enzyme activity drops much lower than 50%.
Interesting. Does that mean AR disorders look different clinically compared to AD?
Generally, yes.
Yeah.
AR disorders tend to have more uniform expression. The severity doesn't usually vary as wildly as we see in AD disorders with variable expressivity, and penetrance is usually complete if you have both bad copies, you get the disease.
Okay. And there's also a connection to consanguinity, right? Marriages between relatives.
Yep. Yes, that's a key indicator, especially for rare recessive disorders.
Yeah.
Think about it. If a specific mutated gene is super rare in the general population, the odds of 2 unrelated people who happen to be carriers meeting and having kids are tiny.
Right.
But if 2 people are related, say first cousins, they have a much higher chance of having both inherited that same rare mutated gene from a common ancestor, like a grandparent. So if a child has a very rare AR disorder, there's a higher likelihood their parents are related.
That makes sense. Now, the AR category is also good for explaining 2 concepts that sometimes get mixed up. Pleiotropism and genetic heterogeneity. Can you break those down?
Sure. They sound similar, but are opposites, really. Pleiotropism is when one single gene defect causes multiple different effects in the body, often in seemingly unrelated systems.
One gene, many problems.
Exactly.
Yeah.
Sickle cell anemia is the perfect example. It's caused by a single tiny change, a point mutation, in the gene for beta globin, part of hemoglobin. But that one change makes red blood cells sickle-shaped and rigid. And that leads to a cascade of problems. The cells break down easily, causing anemia, hemolysis. They clog up small blood vessels, causing pain and organ damage, infarcts. The spleen gets damaged, all from that one initial gene defect.
Got it. One cause, many effects. So it's genetic heterogeneity.
That's the flip side. Genetic heterogeneity is when mutations in multiple different genes can all cause the same clinical outcome, the same disease or trait.
Many different causes, one effect.
Precisely. Take profound childhood deafness, for instance. There isn't just one deafness gene. Dozens and dozens of different genes, if mutated, often in an autosomal recessive way, can lead to deafness.
So if a child is diagnosed with genetic deafness, you don't automatically know which gene is responsible.
Not at all. Finding the specific gene requires testing many possibilities. This is super important for genetic counseling and understanding recurrence risks, because the specific gene involved dictates the inheritance pattern.
Right, okay. Pleiotropism, one gene, many effects. Heterogeneity, many genes, one effect. Got it. Last major pattern, X-linked disorders.
Yep, X-linked. These, as the name implies, involve genes on the X chromosome, and almost all are X-linked recessive.
Which means it mainly affects males.
Primarily, yes, because males only have one X chromosome. They're homozygous for genes on the X. If that one X carries a recessive mutation, there's no second X chromosome with a normal copy to compensate. So the disorder shows up.
And the inheritance pattern is unique because of the sex chromosome.
Very distinct. An affected male cannot pass the disorder to his sons, because sons get the father's Y chromosome, not his X. But all of his daughters will inherit his affected X chromosome, making them carriers.
So daughters are carriers, sons are unaffected by him directly.
Correct. Then, if a female carrier has children, her sons have a 50% chance of inheriting the affected X and having the disorder, and her daughters have a 50% chance of being carriers like her.
Okay. But why are female carriers usually protected? They have one mutated X, too.
That's due to a fascinating biological process called X inactivation, or lionization.
Lionization.
Yeah. Very early in the development of a female embryo, in each cell, one of the two X chromosomes is randomly inactivated. It gets sort of crumpled up and silenced.
Randomly. Like a coin flip in every cell.
Pretty much.
Okay.
So a female carrier ends up being a mosaic. Some of her cells are using the X chromosome with the normal gene, and other cells are using the X with the mutated gene.
And that mix is usually enough.
Usually, yes. Enough cells are producing the normal protein to prevent the full-blown disease. She's phenotypically normal, though she carries the gene.
But not always completely protected. You hear about symptomatic carriers sometimes.
That can happen. Although the inactivation is random, just by chance. In some women, the process might skew heavily. It's in the critical tissues for that particular disorder. Most cells happen to inactivate the normal X chromosome. Then most of the active X chromosomes will be the ones carrying the mutation.
So she might express some symptoms.
She might. She could have a milder form of the disorder, or sometimes rarely a more severe form, depending on how skewed that inactivation is.
Is there a good example of this?
Glucose-6-phosphate dehydrogenase deficiency, G6PD deficiency is a good one. It's X-linked recessive. Primarily affects males, causing their red blood cells to break down, hemolyze under certain stresses, like taking specific drugs.
Okay.
But female carriers, who are mosaics, can still be susceptible. Some of their red blood cells rely on the X with the G6PD mutation because the normal X was inactivated in their precursor cells. So if that female carrier takes one of those trigger drugs, she might experience some level of hemolysis too because a portion of her red cells are vulnerable.
Wow. Okay, so even carrier status isn't always completely silent. That covers the classic patterns really well. So what does this all mean sort of in the bigger picture? How does understanding these Mendelian rules connect to modern medicine and treatment?
Oh, it connects hugely. It's the absolute foundation. That G6PD example leads right into pharmacogenetics.
Using genetics to predict drug response.
Exactly. We routinely screen people, especially from certain populations where G6PD deficiency is more common, before giving them drugs like the antimalarial primaquine. We know from understanding that X-linked enzyme defect that the drug could trigger dangerous anemia in susceptible individuals. That's personalized medicine, based directly on Mendelian principles.
Makes sense. And what about the autosomal dominant disorders? Have those insights led to treatments?
Absolutely. Some of the most impactful drugs developed in recent decades came directly from understanding AD mechanisms, especially those involving cell receptors.
Like what?
Familial hypercholesterolemia, FH. It's usually an AD disorder caused by a faulty LDL receptor gene. LDL receptors are like the garbage trucks on your cells that pull bad cholesterol out of your blood.
Okay.
If you're heterozygous for FH, you only make about 50% of the normal number of these LDL receptor garbage trucks. So cholesterol builds up massively in the blood, leading to very early heart disease.
So how did understanding that help?
Well, it led to the development of statins. Statins work primarily by blocking cholesterol production inside the cell. The cell senses this lack of internal cholesterol and responds by making more LDL receptors. It puts more garbage trucks out on its surface to grab cholesterol from the blood.
Ah, so it indirectly boosts the number of receptors. Clever.
Very clever. And then, building on that, researchers figured out how these LDL receptors are naturally degraded or recycled in the cell. They found an enzyme, PCSK9, that basically flags LDL receptors for destruction prematurely.
Like a bad manager firing the garbage trucks too early?
Yeah, something like that. So scientists developed PCSK9 inhibitors. These are amazing, highly targeted drugs, often antibodies that block PCSK9.
Stopping the firing of the receptors.
Exactly. By blocking PCSK9, you prevent the receptors from being destroyed, so more of them stay on the cell surface, working longer to clear cholesterol. It's a direct therapeutic strategy born from understanding the life cycle of a protein involved in an AD disorder.
Incredible. And are we also seeing connections now between these simple single-gene disorders and the more common complex diseases?
Yes, definitely. The lines are blurring. We used to think of Mendelian disorders as rare and distinct, and complex diseases like diabetes or Parkinson's as totally separate. But now we're seeing overlaps.
How so?
There's really strong evidence now, for instance, that being a carrier for certain autosomal recessive lysosomal storage diseases, like Gaucher disease, significantly increases your risk of developing Parkinson's disease later in life.
Wow. Being a carrier for Gaucher increases Parkinson's risk.
Significantly. Even having just one mutated copy of the GBA gene involved in Gaucher seems to impair the cell's ability to clear out waste proteins, a process called autophagy. And problems with autophagy are known to be central to Parkinson's development. So that silent carrier state isn't entirely silent at the cellular level, and it creates a vulnerability.
That really changes how we think about these carrier states.
Okay, this has been a fantastic deep dive. We've gone through autosomal dominant, autosomal recessive, X-linked, covered the key differences like structural proteins versus enzymes, and tackled complexities like penetrance and expressivity.
We covered a lot.
Yeah.
And hopefully showed how these fundamental patterns are still so relevant, from basic diagnosis to cutting-edge therapies.
Definitely. So what's the final provocative thought you want to leave us with? I guess it's this. While these Mendelian rules are the bedrock, the more we learn, especially about pleiotropism, one gene, many effects, and genetic heterogeneity, what many genes, one effect, the more we appreciate the incredible variation possible, even within conditions caused by a single gene. Think about the Ehlers-Danlos syndromes. They're all disorders affecting collagen connective tissue. But within that group, you see autosomal dominant forms, autosomal recessive forms, even X-linked forms. It all depends on which specific collagen gene or processing enzyme is mutated. The gene determines the inheritance pattern. Absolutely. But the specific mutation, the specific protein affected, and probably a whole lot of other modifying genes and environmental factors, they determine the actual phenotype, what the condition really looks like in an individual. It's elegantly simple in principle, but incredibly complex in reality.